to our biology program. Last episode you studied the flowering plants, but today we are going to complete the story of reproduction by ending it with the reproduction in human beings. Let's go and see what will happen. Reproduction in human beings. As you know, man belongs to class Mammalia, in which the embryo develops inside the uterus till birth. So their eggs are small and nearly devoid of yolk. This class is also characterized with a few number of young due to the parental care. Because of this care, the number of young individuals, they are very small, and sometimes it doesn't reach 1 or 2 of them, and others they are maybe 4, 6. Man as a higher form of living organism and the higher form of this mammalia class, it shows a high degree of parental care, where the young requires several years of raising. So in order to take care of them, it needs a very long time for taking care of them. And this is due to the mental progress that God provided him to be superior over all other creatures. And now, my dear students, to know and understand how we came to life and how we get babies, we can say that we have to study the genital system in living organisms, especially in men. This genital system, it is divided into 2 types. The genital system of the male, and it is called the genital system of male or reproductive system of male. And the other one, it is the reproductive system of females or the female's reproductive system. And now let's start by the structure of the male genital system, in which we found that it is formed of 2 testes, which leads to the tubule of the epididymis, the vas deferens, and the accessory glands, which are the Cooper glands and prostate, till the urethra. It is the end of this system. So another time, this system, it starts by the 2 testes, which are outside the body, and they have a very convoluted tubules. These tubules is called epididymis, and the vas deferens, the tube which join between the epididymis and the other parts, and there is some glands which are called the prostate and the Cooper's gland, and it is ended by the urethra. And now let's go and see the structure, fine structure of this system. And now let's begin by the film, which show the structure of the genital system in men as the following. First of all, the 2 testes. The 2 testes, they lies outside the body in a sac called scrotal sac or scrotum sac. The testes here move out of the body cavity into the scrotum sac during their development within the last months of pregnancy. This position prepares a condition of cooler than the body temperature, which is suitable for spermatogenesis or the formation of sperms. And finally, what will happen if these testes, it is not found into the scrotal sac, or they do not descend down into this scrotum sac? Yes, the spermatogenesis does not occur because it is an unsuitable temperature inside the body, and also this will cause infertility. This will cause infertility. Then what is the importance of these testes? The first of all, this testes, it is important for the production of sperms. The second point, it is important for the secretion of the testosterone hormone, which cause the secondary male characters to appear at the puberty period. In this period, we found that the child begins at the teen ages. We found that it has a very rough sound. It begins to have a beard, or some hair grow into some parts of his body. This is the main characters of the puberty period. And now, if we look for this structure again for the two testes, we found that they are very important in studying them. The first of all, they are lying outside the body. Why? To get a cooler temperature than the body temperature, in order to give the chance for the spermatogenesis to occur. And this is important because it is important for the formation of sperm or the production of sperm, and secondly, it produces the testosterone hormone, which is important for the secondary male characters to appear at the puberty period. And now the second part of this system, which is the epididymis. The epididymis, each testis leads to a group of cumulated tubes called epididymis. The latter fuses in the vas deferens, or this epididymis is connected to the vas deferens, which is the second step or the second part we are going to look for. This vas deferens, it is formed for the transportation of sperm from the epididymis to the seminal vesicle, which is the second or the fourth part after the testis. So we begin by the structure of this male reproductive system by the testes. Then these testes, it was important because they lie outside the body in order to provide the body with the suitable temperature, and this is important for the formation of the sperm during the spermatogenesis process, and this is important for the formation of sperm during the spermatogenesis process. The second part after that, it was the epididymis, and this epididymis, it is a cumulated group of tubule or small tubes that convolute in over each other in order to let the chance for the formation of this sperm inside it. This epididymis join between the testes and the vas deferens in the second step after that. The vas deferens transport the sperm to the third or the part which is found after that, which is called seminal vesicle. The number of seminal vesicles, it will be 2 also. So we have 2 seminal vesicles. These seminal vesicles were very important for the sperms, where the sperms are stored till they leave the body. These sperms which are stored inside it, they are stored until they are needed. That's why they are kept inside these seminal vesicles. After that, there is something which is very important, which we call it accessory glands. The accessory glands here, they are 2 accessory glands, the prostate and the Cooper's gland. The prostate here, they are important, and also the Cooper's, they are important because they secrete an alkaline fluid to neutralize the acidity in the urethra. Why? Because the sperm can't live inside an acidic medium. That's why this acidic media, it is unsuitable for the sperm, so God creates these glands to provide the alkaline fluid in order to neutralize this acidity and make the medium neutral, so the sperm can live and pass through it through the urethra after that. Then we came to the final part of this system, which is the penis. The penis here, it is a spongy tissue, a very soft one. And this spongy tissue is a part of this organ. The penis contains the urethra, where the sperms passes through it, and also the urine, but individually. That means when they are expelled outside the body, each one it is expelled alone in different times between them. First, why? Because the urine, it is a very acidic one, so the sperm can't live inside this acidic medium. That's why they are, after the urine passes, we found that the Cooper glands and the prostate secrete their fluid, and this fluid, it neutralizes the path of this urethra, so the sperm can be expelled safely. Now, my dear students, we knew the structure of this genital system, or the male genital system. What about the testes? What if we look inside these testes? What we are going to find? The testis is built up of a seminiferous tubule. These tubules, they are very small, and in between them there are another types of cell which is called interstitial cells. These interstitial cells, they secrete the testosterone hormone. Look inside this tubule of the seminiferous tubule. We found inside each tubule there are Sertoli cells. These Sertoli cells, which secretes the fluid to nourish the sperm inside the testis. So if we go back and again and repeat this part, the seminiferous tubules are very small tubules. In between them there is the interstitial cells. Why there is interstitial cells? In order to provide the testosterone hormone, or to secrete the testosterone hormone. Also, if we look inside each tubule, we will find the Sertoli cells, which provide the nourishing fluid for the sperm. All of this, it is inside the testes. Also, the testes provide an immunization function, which is provided from these types of fluid and these types of secretion from the testes. Each tubule is lined internally with primary germ cells. Be careful and pay attention for this part. We are going to study the spermatogenesis, and in order to know the spermatogenesis, so we have to revise this part. Each tubule is lined, the seminiferous tubules is lined from inside by a primary cells which are called germ cells, and they are diploid. That means they contain 2 N chromosomes. They are divided and finally transformed at the end of these stages. They are formed at the end into sperm, changed into sperm. Then, what about the sperms? How they are formed, and the beginning of this formation, and the process of this formation, what it is called. We call them this process, we call it spermatogenesis. So the phases here of this spermatogenesis, they are divided, or they are split into 4 types, or 4 main phases. This process by the 4 important phases, which are the first, multiplication phase, the second, growth phase, the third, maturation phase, the fourth, metamorphosis phase. Come and follow the formation of this sperm as you remark in the following film. The first stage, which is the multiplication phase. In this phase, we found that the we found the primary germ cells which are diploid. We found that they begin by a division which is called mitosis division, and this is a type you found that it makes typical cells for the mother cell or the primary cells. So all the cells produced here, they are diploid. The primary germ cells, they are diploid. As a result of this division, a great number of spermatogonia cells which are diploid are produced. So the first one, the multiplication phase, in which the germ cells are multiplied by a mitosis division and form the spermatogonia, which contains 2N chromosome, or they are diploid. Then we will go to the second stage, which is the growth phase. In this growth phase, we found that the spermatogonia begins to store some amounts of food and are transformed into another phase which is called spermatocytes. So the growth phase, it is in which the spermatogonia store an amount of food, and this food for feeding the sperm and provide the sperm with nourishing substance needed, and are transformed into the second phase, which is spermatocyte. The third phase, it will be maturation. The maturation, it is the stage where the spermatocytes are called primary spermatocytes, which are diploid, as you know. They undergo into another type of division, which is called meiosis division. And this meiosis division, it takes place on two steps. That's why we call it, the first one, we call it meiosis 1. And as a result of this meiosis 1, we found that there is a spermatocyte, or what we call it secondary spermatocyte, which is an haploid cells. So the spermatocytes, primary spermatocytes, contains 2N chromosome, or they are diploid. And after the meiosis 1, which takes place in this cells, they begin to form the second stage, which is the size haploid. Then this secondary spermatocyte undergo a meiosis division, but this is the second stage. That means the second step of meiosis, in which there is a replication takes place, or a greater number of cells is produced. As you know, in the first secondary stage, you studied this one, and you knew that the meiosis division produces at the end 4 cells. So these 4 cells, each one it is haploid. So if we go back to our secondary spermatocyte, which is a haploid one, and they are 2 cells, these 2 cells begins to undergo to the second meiosis division and produces the cells which are haploid spermatids, which are N chromosomes, or contains N chromosomes. They are haploid. The number of chromosomes here, it is reduced from the 46 chromosome to the half, which is 23 chromosomes. So at this phase, there is 2 things happen. First of all, the primary spermatocytes undergo a meiosis division, which takes place on 2 steps. In the first step, the secondary spermatocyte is formed, which is haploid, and in the second step, or the meiosis 2, the spermatids are formed, which are also haploid, and they contains, each one of these spermatids contains 23 chromosomes, or the half number of chromosomes in men. The last phase in this spermatogenesis, it is the metamorphosis phase. In this phase, the spermatids are converted into sperm or changed into sperm. Let us revise this part, or the spermatogenesis. In the multiplication phase, we found that in the first one, the mitosis division takes place for several times, and the primary germ cells, it is diploid, and as a result of this division, a great number of spermatogonia cells which are diploid are produced. The second phase, it is the growth phase, in which the spermatogonia store an amount of food and becomes a little bit larger. That's why we call it growth phase, and they form spermatocytes. These primary spermatocytes in the maturation phase, they begins to change from being diploid, undergo meiosis division 1, and form the secondary spermatocytes, which contains a haploid cell also, and they undergo a meiosis division second step or second stage, which form the resulting haploid of this haploid spermatids, which is resulted from this phase. It is from the maturation phase. Also, we knew that in this maturation phase, the spermatids contains the half number of chromosomes in the man, which is 23 chromosomes. The final phase, we found it the metamorphosis phase, in which the spermatids are converted into sperms. Then what is a sperm? And what is the structure of this sperm? What is its function? Why it is very important for us? As we look for this figure, we found that the sperm is consisting of 4 main parts: the head, the neck, the midpiece, and the tail. Let us see the structure of this sperm in details. The first, the head, which contains the nucleus, which contains 23 chromosomes, and there is a very important part which is found into the forehead or at the tip of this head, which is the acrosome. Why it is important, this acrosome? This acrosome secretes hyaluronic enzyme, which dissolves the ovum membrane, part of it, and this facilitates the entrance of this sperm to the ovum and form the fertilization process. The second part of this sperm, it is the neck, which contains 2 centrioles that play an important role in fertilizing the ovum division, which play an important role in the fertilized ovum when it divides, or the division of this fertilized ovum. Neck, it contains, as we said before, the centrioles, 2 centrioles, and they are important for the division of the ovum after that, but after being fertilized. The third part of this sperm, it is the midpiece. The midpiece, it is the generator of this sperm, where the midpiece, it contains the mitochondria. This mitochondria supply or provide this sperm with the energy needed for its motion in order to travel from the whole genital system of females. Then at the end of this part, it will be the tail, which consists of an axis which ends with a caudal. So the tail consists of two parts, which are the axis and the caudal part. So the caudal part, it is very important in order to help the sperm to swim and move till it reaches the ovum. So another time, the structure of this sperm, the sperm, it consists of a head which contains the nucleus, which contains the 23 chromosomes, and the acrosome, which secretes the hyaluronic enzyme to dissolve the part of the membrane of the ovum and help in the penetration of this sperm inside it. The second part, it is the neck, where we found the two centrioles, which play an important role in the division of the fertilized eggs, ovum, the fertilized ovum, and the third one, which is the middle piece or the mid piece, which contains the mitochondria, and these mitochondria provide the energy for the sperm and help it to move easily. Also the tail, which is the final part, which consists of an ax and the caudal part, and this help the sperm to move till it reaches the ovum. The final point of our program. Thank you for watching us and see you next time.